All right, so it's time to begin combining different fields of knowledge. In the past, we studied cells. Now we're going to see how do these cells relate to genes. So we've seen this before. Normal Punnett square cross of two purple flowers that are heterozygous. But how do we go from genes that are coded by DNA to a physical trait? That's our topic for this video. First off, do you remember cell structure from last year? Where we had the nucleus in the middle where the DNA is stored, mitochondria, the powerhouse, vacuoles that store things, the basics, right? We're going to focus just on the nucleus. Right? This is where the DNA is stored. The DNA never leaves the cell. These are two videos I'm going to ask you to watch later on your own time. I'll post these slides to Classroom so you can access these. These show visuals and basic explanations of how DNA is copied out and becomes a protein later on. Turns out it's proteins that guide most of the actions of a cell. So genes, to the simple coding, and the phone. That I was saying. So genes are coded by for DNA within the nucleus. They're copied out as RNA, which is just a transfer molecule. And then these are translated to become proteins. And it's proteins that actually do the work in a cell. DNA just has the instructions. But what if something goes wrong? Something could go wrong when traits are passed on to the next generation or in the life process of a cell. All right, so what happens when things go wrong? When information is corrupted, the DNA is altered when at conception, when a new individual is formed. So here we have the normal heterozygous cross. But what if something happens with the female plant and the recessive B gets altered? I don't know how it's going to be altered, so let's just call it B prime. So this gene is different. There's some mutation, there's some change in the DNA. This is a mutation. Even a small change can be very dramatic, very harmful in the case of some species, especially in humans, especially in more complex animals. So in this case, we don't know what this B prime trait is going to do. All we know is that because the DNA has been changed, the RNA transfer molecule will also be different, and the protein that it makes could be changed as well. We've seen this. Sickle cell trait is a mutation of the normal red blood cell. Right? So that's why mutations can cause dramatic changes in what's going on in the cell. Right? So to recap, DNA codes for genes, DNA is copied into RNA, RNA is translated to a protein, proteins do the work in a cell. It's important to see the connections then between the cell biology we studied in the past and the current genetics that we are studying now. Understand how change in the DNA and the sequence of letters that make up for a gene can have huge impacts downstream.